Me okay? Great. All right. So what I'm going to do is I'm going to start off with a video that Brooklyn made, and this was shown at a session organized by the World Bank at the Global Landscapes Forum yesterday. And they asked her to put this video together. She had about 2 days to do this, so I was so impressed. So yesterday we gave this talk at this session, which will reach thousands of people. Actually, started with the video, and then I gave a talk about bat research. So I'm going to give a similar talk now, but keep it more focused on what the lab does and what our experiences are like working in these systems. And then Brooklyn, Dale, and Will are all going to give a short presentation on what they are doing in the lab. And then we're trying to leave lots and lots of room for questions, because I think questions are always the most fun part. So I don't know, do you interrupt me during the talk or do you wait till the end? I don't mind either way. If you want to interrupt me with questions, you can go for it. But I'm going to start with the video here right now. All right, you should all be able to see this.
My name is Brooklyn Hunt, and I'm an undergraduate here at Montana State University. The Plowright Lab is one lab within a global consortium of many labs known as Bat One Health. In Bat One Health, our goal is to understand the spillover of bat viruses and how that changes based on ecological landscape changes and land. Within Bat One Health, we have collaborators all over the world. But specifically, fieldwork sites where bats are actually caught and studied in Ghana, Bangladesh, Madagascar, and Australia. We specifically study Hendra Virus in Australian flying foxes because it's really just an ideal system to understand spillover. So in order to do that, we study bat immune function, microbiomes, hematology, diet ecology, foraging behavior, and lots of other things. But specifically, my research examines the diets of black-flying foxes because we're interested in how changes in habitat alters their diet composition and how diet change affects spillover. I also do a lot of hematology, which is the study of blood, because I want to expand our ability to gather information on bat health from blood samples. At the level of Nepahvirus itself, we study how stable the virus is in different environmental conditions, specifically how stable it is. So I'm going to hand things over to Dr. Raina K. She's going to tell you more about our work on Hendra Virus and how we can apply our work and our findings to help stop bat virus spillover on a bigger scale.
Yeah, that was the opening of our World Bank session at the Global Landscapes Forum. Isn't it fantastic? I was so amazed when Brooklyn showed that to me, that she could do that so quickly. And I'll discuss in a minute why you saw lots of bats drinking mango juice and being released. Okay, well, actually, I'll tell you right now why that is. Because we work on pathogens that are really, really dangerous to humans. So these are the pathogens we work on are biosecurity level 4. So that means that if we wanted to handle these viruses, we'd have to take them into a lab, the BSL-4 lab, where you have to wear a spacesuit, you have to have an external air supply, and then you have basically the labs are set up where you're a box within a box in a box with extraordinary security clearance to get in there. So we're working in the field with full PPE. We're at much less risk than when we actually have the virus, but we have to be really careful about how we're perceived when the community sees us working in their communities on bats. And so when we show videos, we don't show a lot of videos of us in full PPE because we don't want to scare people. And we don't want people to get scared of bats too, because bats really, they're not the enemies here. They're the canaries. I'm going to explain that too. And then the other thing is we're handling animals. So our work involves catching animals, giving them an anesthetic, and taking samples. And there's a lot of controversy whether that's appropriate. Some people think it's not appropriate to catch a wild animal or handle a wild animal. And so we when we show social media or anything on social media, we just tend to show bats drinking mango juice or being released, because you really, no one can say that they can't enjoy watching a bat drinking mango juice or flying away from a human. So that's why we don't show a lot of detail there. But I am actually going to show you a little bit more in my talk that we wouldn't have shown in a large public forum like that. So I'll just start sharing my screen here. Okay, can you all see this okay? Okay, so I'm showing you here, this is Tropus, Tropus medius. This is a big fruit bat. We call them flying foxes too. This is the host of Nepahvirus. Nipah might be a virus that you've never heard of, although there's a lot more discussion about it these days. But I'm going to talk a little bit about this virus. And of course, I just want to acknowledge this incredible team of people. We have over 70 people working on this project. I think we actually have more because many people are missing from this older series of photos. And what we're really trying to do is trying to understand how pandemics begin. We're really focused on the viruses that come from bats, and we're wanting to understand all the factors that allow those viruses to jump into humans. We want to be able to predict these events. We want to be able to prevent them. Events just like SARS-CoV-2, really, which was a bat virus, and it was happily living in the intestines of a bat until, gosh, what are we, like 11 months ago or so. And this is, SARS-CoV-2 Nipah virus. They're one of many, many emerging infectious diseases, and we understand there's this acceleration of the diseases coming from animals into humans. But so often we're seeing these new diseases coming in from bats. And here again, another bat drinking mango juice because, I mean, really, can you get enough of these bats drinking mango juice? They're just so cute. This is a Pteropus electo, the black flying fox. Brooklyn mentioned the black flying fox in her video a minute ago. These are the major reservoir for Hendra virus in Australia. And these guys, as I said before, they're not the enemies. Bats perform such critical ecological services that no other species can perform: pollination, seed dispersal, insect consumption. And they're really just an indicator of our relationship with our environment having gone wrong, that we're seeing so many viruses come from bats at this point in time. So I'm going to run through now some of the science of what the kinds of things we're working on in the lab. One is to try to understand how pandemics start. And this is just going back to the real basics here. For a pandemic to start, and we're talking about zoonoses, so these are viruses or pathogens of any sort that come from animals and then are transmitted to humans. So for a zoonotic pathogen to ignite into a pandemic, we have to have an infected reservoir host. In this case, we're thinking about bats, but it could also be, livestock, it could be rodents, it could be many, many different species, birds, pigs for influenza viruses. So we have to have infection here. Now, how these infections move within wildlife hosts is really complicated. So that's a big part of what our lab does, is trying to understand how these viruses transmitted from individual to individual, population to population. Why do we see big highs of high prevalence, lots of animals infected, or troughs in prevalence, not many animals infected? Then that pathogen, it's got to get out of that host. So it has to have some sort of exiting of the host. And that could be, say, through shedding. Like here, the viruses that we work on, the Nepahviruses, they're shed in urine and or coronaviruses and feces. But it could also be from a bite, from a vector. It could be through slaughter for bushmeat consumption. And then the virus has to get into the next host. And we call that spillover. And that could be through direct contact with the pathogen or with the host. And there are many complexities there. It's actually pretty hard for a pathogen to go from an animal to a human. Most of the time we're exposed to pathogens from animals and we never get sick. And that's because we've got lots of barriers, got a robust immune system. Many of these pathogens couldn't bind to our cells. They can't really replicate in our cells if it's a virus or be disseminated through our body. There's a lot of things that these new pathogens have to overcome for us to be able to be infected. And then that virus is then going to be able to spread in the community. And that's actually kind of hard to, actually what we work on with the Nepahviruses, they don't spread very far. So far, all the viruses are spilled over haven't spread very far. But if a Nepahvirus started spreading, I'll explain soon, that would be pretty tragic, much more tragic than SARS-CoV-2 potentially. And then many of those processes that trigger these pandemics, they happen in the context of a landscape. And the trigger for these processes is often some kind of landscape stress, like deforestation, loss of critical habitat, processes that force animals to come into contact with humans or force animals to be in a condition where they're shedding more pathogen. Often with more human population density, we've got more spread amongst humans. And so that's where I'm going to introduce this concept of landscape immunity. This is really a new concept, and this is thinking about how could you prevent infect, shed, spill, and spread? How can we prevent that by managing landscapes so that animals are healthy? We don't see animals all aggregated in one area with high prevalence of disease. So why animals don't have to come out of their habitats to contact humans and so on. I'm getting an annotation request here. I don't know what that means, but I assume I just declined that. Okay, let me know otherwise if that's not the case. I have a link here to a preprint on this concept. Okay, so what about what we're dealing with right now, the COVID-19 pandemic and this infect, shed, spill, spread process I've just described? Was that relevant? And were landscapes relevant? Well, absolutely they had to be. They had to be relevant because the reservoir hosts of COVID-19 or SARS-CoV-2, is what we call the virus, they're horseshoe bats, Chinese horseshoe bats. And the closest relatives to SARS-CoV-2 have been found in Chinese horseshoe bats in this area here. Can you all see my pointer? Good, okay. So, northern Vietnam, Laos, Thailand, Myanmar, and Yunnan province, southern China. This is an area of very intensive land use change. So fragmentation, deforestation, also very high population density and high livestock density. So lots of people, lots of livestock, lots of opportunities for bat-human proximity. We don't exactly know how the virus got to Wuhan, but certainly there had to be an infected bat somewhere in here that somehow that virus has gotten out of the bat into probably an intermediate host that was taken to Wuhan, or perhaps even a bat was transported to Wuhan. But there was a bat-human interaction at some point that is certainly, and this whole process is often triggered by more humans on the landscape, more humans intruding into bat habitat or bats having to come out of their habitat to find food. So how do we actually predict this process? Well, that's really tough because if you think about coronaviruses, we've had 3 that have emerged into humans in the past 20 years. So this current pandemic, we've got Middle East Respiratory Syndrome or MERS that actually went from bats to camels, and then from camels it goes into people usually in the Middle East, and then the first SARS outbreak, which was in 2003, which went from bats to civets to people. But all of these are probably just a single spillover from bats into this other host. And so with that, like n equals 3, it's very difficult to build up the kinds of patterns to understand how spillover happened and then how we can stop it from happening again. And if you look at the map and you look at the coronaviruses, this is work from my lab and the whole Bat1 Health group actually as well, thinking about where bat coronaviruses exist, and they are throughout. You can see the very wide geography, 3,000 bat coronaviruses, probably a couple of hundred that could infect humans. So if we want to prevent SARS-CoV-3, we need to really think about how we could do that, given we have little data to understand how these last few have happened. So what we want to do is then we want to go to systems where there is a lot of spillover, and we can start to put the data together to understand the patterns of spillover. And when we can understand the patterns and the mechanisms driving spillover, then maybe we could figure out what to do about it. So we work in, amongst other places, we work in Bangladesh, This is Nepahvirus, this is space, this is time, every dot. This is east coast of Australia, this is time, every dot is a spillover event. So we've got a lot of data, relatively speaking, for these emerging diseases. So we're trying to put this together to understand how it all happens. And we're back to Pteropus medius, the very beautiful fruit bat host in Bangladesh. So I'm going to start with Nipah virus. Nipah emerges in this context every year, spillover events in a highly densely populated country with a very poor public health infrastructure. It's a WHO priority 10 pathogen, as is Hendra. So that means no countermeasures, no medical vaccination countermeasures for this disease. Nipah has a fatality rate during outbreaks between 50 and 100%. So often it's around 70, 90%. There were 2 outbreaks in India over the last couple of years. They had a fatality rate around 90%. So far, the virus only goes through very short chains of transmission in humans, but the worry is if the wrong genotype of virus gets into humans and it can spread effectively, that we would be in for a civilization-changing event. I mean, could you imagine SARS-CoV-2 with a 50 to 70% fatality rate? It's unimaginable. So Nipah virus it gets into humans through the spillover process when people collect date palm sap. So here's someone making a lash in the, they make a big gash in the tree, and the date palm sap drips into this pot, and then it's sold on the street the next day. And this is video from a colleague of mine who works in this system. He's a bat licking the date palm sap. And if you watch really carefully, the bat will turn upside down and pee right in that date palm pot. And you can track the spillover events by actually following the date palm seller's tracks. So here's a date palm seller's track. He walked along this road here in red, and the spillover events are the red dots here. So these are people who got sick from Nipah virus, who are on that date palm seller's route. But I'm actually not going to talk any more about Nipah virus. Nipah virus is happening in a very transformed landscape, one was deforested decades ago. I'm going to talk about Hendra virus in Australia. So it's the relative of Nipah virus. It's in the same genus within the family Paramyxoviridae. Hendra is naturally occurs in the flying fox, big fruit bat species. They're nectar feeding species, even though we call them fruit bats. They love nectar, but they'll eat fruit when there's no nectar available. If a bat feeds in a tree in a horse paddock and urinates or defecates on the ground, and that horse then consumes that contaminated grass, horse can become sick. Horses have an 80% fatality rate. They get very sick, and they amplify the virus to really high levels. And then humans get infected from horses when they treat sick horses. And now any veterinarian ever dealing with a horse on the east coast of Australia, if that horse has a fever, respiratory signs or neurological signs, you have to have full PPE. And it's become actually an absolute nightmare for the veterinary industry in Australia. But what we've discovered in Australia is that these spillover events actually happen in clusters. So I was talking about trying to build patterns so that we could figure out what causes these. Well, we see 2011, a bunch of spillover events, but then nothing till 2013, a few in 2014, and then a cluster in 2017. I'm going to take you to 2017. We actually went onto the ground to one of these spillover events, one of these dots here in Australia. And we talked to the owner of a horse who just died of Hendra virus just recently. And she described what happened. And I had my whole team there. We just got an NSF grant. We went to Australia. We were studying this system. And as she described what happened to her horse, it was this amazing experience because we'd just come up with this conceptual overview of how spillover happens, that to get from for a pathogen to go from an animal to a human, or here actually from one animal to another animal, it has to cross all these different areas. All these things have to align in the same place and time. So she described it was raining, her horse's paddock turned to mud, so she put her horse into the lemon orchard. Recently, some bats had moved into a property very close by to her property, just about 1 kilometre away, less than a mile away. Because those bats were experiencing at that time a starvation event, there was a huge acute food shortage. The bats came into her orchard where she put her horse to feed on lemons, which are a food of last resort, starvation of food, avoidance food. Because those bats were starving, we now understand there's a link between nutritional stress and shedding virus. So those bats were shedding virus, and because it was cold and wet, there was really nice conditions for virus to survive. So as those bats excreted urine on the ground while they were feeding the lemon trees, that virus then was able to survive in the grass for hours, for long enough for the horse to come along and get an infectious dose and then be buried, and for her family to go through the most stressful event of their life. Thankfully, no people were infected. But it really put this whole framework in context that you had to have reservoir hosts there, they had to be infected, they had to release that pathogen, pathogen had to survive, and then the new host had to be susceptible. So it had to go through all these within host barriers. And we had actually predicted, though, that cluster in 2017 because we knew that the bats were going to have a food shortage then. And we knew that because we've now understand the climatic cycles that drive these food shortages. This is the El Nino cycle. We knew it because the bats had really low reproductive success. And we'd also seen a lot of bats come into rehabilitation centres. And we now know when we see all of these things together, we are going to see Hendra virus in horses in the following winter. And why winter? And why are we seeing, how does this all start? This all starts, we understand, from loss of winter habitat of bats. So in summer, many trees produce nectar, there's lots of food. In winter, the bats, there isn't any, basically virtually no winter habitat left. So very few species actually produce food. In winter, they've almost entirely been cleared. So they get to winter, if there is climatic oscillation, there is an acute food shortage, and the bats split up from big populations feeding on nectar to small populations in agricultural areas near horses. And this is what it looks like. It looks like this proliferation of bat populations. So this is in 2002, the red dots are bat roosts, and in 2017 there are now bat roosts across a much wider distribution. And we understand, I'm showing this video again because I just love this so much. This is from one of the PhD students, Adrienne. This bat has a collar on, and now we know, and this is work actually by Maureen Kessler in my lab, when those bats are starving, they're feeding in horse paddocks on weedy species. And this is some of the work that Brooklyn and Dale are doing, showing what their bats are eating, showing the bats are eating crap food, really crap food when there's a starvation, when there's this food shortage event. But when there's nectar, the bats will fly right over that food to nectar. So basically this because of this loss of habitat and this splitting up into small populations in places where horses live, we're getting this contact with horses, so increased contact between bats and horses. But we also know we're getting increased viral shedding. So these bats are more likely to be infected, and they're more likely to be having that pathogen released in their urine in a horse paddock. And we know that, and I don't want to take too much time here, but I just wanted to show you more of the video that was, you saw a bit with Brooklyn's video of what we do. And Brooklyn was supposed to be in Australia right now doing this, but she'll tell you a little bit about why she isn't. And I think you could probably all guess why she isn't. So we catch them in this big mist net as they're coming back from their nightly feeding. And they fly right into the net. We bring the net down, and then This guy's actually eating a weedy species, a very poor quality food there, crocus palm fruit. We put him into a pillowcase. Then we actually remove a tiny, it's a residual tooth. And it takes about 3 or 4 seconds to pull it out. And from that tooth, we can get the age of the bat. We actually do an analysis of the cementum of the tooth. It's like tree rings. We can actually count the tree rings on the tooth to get the age. And age is really precious for understanding disease. We take blood. We do, in our lab, we have a lot of immunology going on. We do look at the bacterial parasites within the red blood cells. We look at a whole bunch of different parameters that are related to the condition of those bats. This is taking the urine there because that's where we actually find the virus. And you've seen this, of course, with the release there. And that's easy. That's in the middle of a public park where many bat populations live now because they're now occupying more of the urban areas. But it's not that easy all the time. So this here is in the Northern Territory of Australia, a very remote part of Australia. It took hours of driving on dirt roads to get here. And this is a population of hundreds of thousands of little red flying foxes. And we were working on the banks of the river catching the bats because they'd love to roost along rivers. But the crocodiles were starting to learn our patterns, of the big saltwater crocodiles there. So we had to find other ways to be safe. So we did this on a boat. And it was great. We could work during the daytime on the boat and catch the bats in a very similar way. And we thought we were very clever until the wildlife national parks caught a crocodile that was longer than our boat on the bank of the river where we were launching our boat every day. So there are some challenges in doing field work for sure. Okay, and then all that field work, which is months of work, getting up at 2 a.m, catching bats. It's very, very hard work. What that gives us is then data. And here's data aggregated over 5 sites across Eastern Australia. Each dot is the proportion of bats that had the virus. And I'll just take you to this dot. That's that bat population near the horse that I just described. Over 60% of the urine pools that we collected from bats then were positive. Let's talk about prevention and prediction. We actually, like we predicted 2017, we then predicted again 2020, same conditions. We're going to have spillover events in horses, so we put warnings out. And we did actually get a spillover event in early June, which is very early because winter in Australia obviously is when our summer is. And we thought, oh gosh, it's going to get very bad. But then it stopped. There were no more spillover events. And now what we understand is that there was this massive blooming of eucalypt flowering. So one of the forests that only flowers maybe every 5-7 years had a big flowering event in winter. And so in July, here's a population here, 5,000 bats were roosting here. Then that's increased to 200,000 bats roosting here. And they were flying out from here into the forest surrounding where there was a big pulse of nectar. Actually, 70% of one of the species of bats, Tropus polycephalus, went to this location. And what we think is that stopped the spillover event. And now we understand that whenever there is a big bloom of winter flowering, we don't see spillover. So this means Hendra Virus. So 0, 0, 0 when we see, for example, the spotted gum flower. That's one very sporadic flowering, but big stands of spotted gum, but they don't flower very often. So we're actually now working with the Australian government on this countermeasure, which is restoring that winter habitat. And we think if we restore those trees that provide food in winter, we actually stop this whole process happening. Stop the fissioning of bats into small groups and stop spillover because bats will have enough to eat in winter. But the only way we've been able to do that, this is my last slide here, is that we've got a lot of data over a long period of time. So we really understand the system. We get to the root cause of what is causing this spillover. And then we can then reverse, we think, the spillover by, again, coming back to this concept of landscape immunity, restoring that landscape immunity so bats can do what bats do, without having to come into contact with livestock and humans. And thank you. I'm going to hand it over now to Brooklyn. Brooklyn, I'll just advance when you let me know.
Okay, that sounds good. Can you hear me?
Yes, I can hear you.
Okay, perfect. Well, hi everyone. I'm Brooklyn Hunt. I'm the one that you just saw in the Global Landscapes Forum video. I am here today to tell you some of the adventures I've had as an undergraduate researcher with the Plowright lab. Like Raina was saying, I'm going to tell you kind of everything that happened at the beginning of the pandemic in my life. And then I'm just going to go over a little bit of what I've been working on since I didn't get to go to Australia. So to start out, I started my undergrad research career in fall of 2019 studying variations in diet composition of black flying foxes. So that picture on the left right there, that is a black flying fox. And then over on the right, we have several panels of just some example diet items of what a black flying fox might eat. So I was really excited to be doing this research, fell in love with it, and I decided that I wanted to grow more research skills and I wanted to travel to Australia and actually do the field work for a study. So I put in hundreds of hours planning a study abroad. Actually, Dr. Schultz from the Honors College was huge in kind of helping me plan a lot of things, and then applied for tons of research grants, all this stuff. I was set to leave in May, and around March, we kind of started going, oh, COVID's not looking too good. And we expected, that international travel was going to get shut down pretty fast. So I made a pretty fast executive decision to just leave in March and finish my semester at MSU online, and just get down there so that I could help out with field work, especially because we were expecting it to be such a bad year for Hendra Virus. So at that point, when I made that decision, I had no visa, I had no international credit card. I was on crutches at the time because they thought that I might have torn my meniscus. I had not secured any grant funding, and I had so many things just working against me. I was pretty much constantly emailing Raina and our Australian collaborators just every second of the day for about 2 weeks. So we got to, almost got there. I was set to leave Saturday morning, fly out on Saturday morning, and Thursday morning before that, I wake up, check my phone, and right there, it says that my flight to Brisbane is canceled because they are shutting the border on Friday night. So Raina, you can advance the slide to that photo of Brisbane. That is the beautiful city that I had been planning to go to. Great city, super excited to experience the culture and do research, and I was just totally crushed. So I took a few days to feel sorry for myself and then realized I needed to figure out something to do now that I was going to be in the US. My diet study had pretty much been put on hold because we couldn't get any samples from our collaborators who were processing them, making sure there was no Hendra virus in there that could infect us. So that was on hold. I was looking for something else to do, and I ended up on this study of flying fox blood. So Raina, if you could advance the next slide. So right now, we're still working on this project. This is actually a project that Dale and I are working on together. So I'll introduce it and then turn things over to Dale to finish. So why are we doing this work? Well, like Raina said, it's really important to understand kind of like holistic bat health in order to understand spillover. So we've been gathering lots of blood from these flying foxes every time we catch them down in Australia. And we're taking blood slides and looking at the morphology of the blood cells on those slides. So we're focusing on 4 main white blood cell types. If you know a little bit about immunology, you know that white blood cells are immune cells that fight infections, do tons of different things. So we're focusing on eosinophils, monocytes, neutrophils, and lymphocytes. We don't have any basophils on here because it seems like bats don't have very many basophils. We also look at some red blood cell morphology, etc., but that's a little too complicated to go into right now. So I don't want to go through all of the results that we've found so far, because we don't have an hour. But I'm going to show you pretty much the most exciting result we've gotten so far. So Raina, if you could advance. Perfect. These cells probably don't look super exciting to a lot of you if you've never done hematology before, but they're really exciting for me. So these are lymphocytes. These are granular lymphocytes specifically, and even more specifically than that, they are cytotoxic or natural killer T cells. So if you've taken immunology, you're probably going, ooh, right now. I'm not going to go into what these cells do. I'm going to let Dale do that. So Dale, I will hand it over to you now.
Okay, awesome. Can everyone hear me? Okay. Yeah, so these natural killer cells that we're seeing were, as Brooklyn was saying, they're really exciting for us. Natural killer cells are a unique population of immune cells that are present in most mammals. They're innate immune cells, which means that rather than having a specific receptor for a particular antigen, like a B cell or a T cell might, they're able to respond to a really wide variety of pathogens. And they're able to do that very rapidly. So activated natural killer cells are associated typically with the beginnings of an infection, usually a viral infection, but they can respond to other types of pathogens as well. But the reason why we were surprised to see this in our sample is that many species of pteropid bats lack canonical natural killer cell receptors. So in Pteropus electo specifically, that altered receptor repertoire means that the activation threshold for these natural killer cells is much higher than you would typically expect. So the fact that we're seeing these activated cells means that this particular population of bats might be experiencing a really intense pathogen challenge. So we're interested at looking at what virus or parasite these bats might be infected with. And Raina, if you could advance the slide. So I've been involved with the Plowright lab since 2019, and I've been able to do a lot of projects. Currently, my research is focused on looking at those factors that contribute to spillover events and contribute to viral shedding from these bat populations. And as Dr. Plowright was saying, one of the biggest factors is nutritional stress. So when bats are experiencing when bats are experiencing a lack of available nutritional resources, they're more likely to shed viral particles and trigger those spillover events. So one of my projects is looking at Pteropus electo, which is the only flying fox species that has been associated with Hendra spillover events thus far, and comparing their diet preferences to a related species, Pteropus poliocephalus, pictured on the left there, to see if they differ in how they utilize food resources in the context of their availability. So Dr. Plowright pretty much explained the significance of that. When we're looking at that, we're trying to understand if these bats might have preferences that could drive them into closer contact with human or horse populations and thus trigger spillover events. The other project that I'm working on is pretty closely related to that, and that's looking at ketogenesis in bat populations. So ketogenesis is a metabolic pathway that's used by many different species of mammals, and the purpose of ketogenesis is basically to generate ketone bodies, which are able to cross the blood-brain barrier to provide energy for the brain in low glucose conditions, so under situations of nutritional stress or starvation conditions. So this is significant for bats because flying fox bats and old world fruit bats in general actually lack a key gene, the HMGCS2 gene, which encodes for a critical enzyme in that metabolic pathway. Knockout studies in mice have revealed that individuals that lack that gene are unable to engage in ketogenesis, meaning that they're especially sensitive to starvation. So that has the implications for Hendra virus spillover then, is that bats that are unable to engage in ketogenesis might then be especially sensitive to starvation conditions and thus more sensitive to a lack of food resources. More recently, my work in the Plowright lab has been involved with the COVID testing, which I think Will is going to talk a lot more about. So I'm going to turn it over to him.
Cool. I have only really briefly been involved with the Plowright lab. I typically work with like elk and wild dogs and lions, tigers, bears, that kind of thing. But I got involved, late in the summer, just on this COVID modeling project where we've got this issue where we've got limited tests, we don't know where to put them, and we also don't know how students might react to testing. And so I spent a solid 2 or 3 weeks like coding day in and day out these compartment models, which basically are a theoretical overview of infection. So when you get the sniffles from a cold, maybe you can envision you're susceptible before you get infected, and then afterwards you're recovered from that individual, cold or whatever it is. And we can take that same theoretical overview and make it way, 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 way more complex. So in this model, we were thinking about the student population with kids on campus, kids off campus, and faculty, and having disease flow in between these three subpopulations. And then also thinking about how kids would come get a COVID test and what those results would mean for maybe when the school should shut down, if it should shut down, how many tests per day might be required, and if testing itself could be useful in limiting the epidemic. Now we're kind of progressing into this LAMP universe that you might have heard about in the beginning, where we've got this really rapid test now where you can basically, it's a saliva-based test, so it's far more efficient and probably less prohibitive to the patient than like a nasopharyngeal swab. But it's unsensitive. So it means that it can't always classify a true positive. And so now we're trying to think about how to optimize those conditions, how to retest individuals, maybe most optimally, how to pool, and then also incorporating some of those disease parameters, like how often students are going to actually show up to a clinic to spit into a tube, how many times to retest an individual student, how isolation might work, how many more isolation rooms MSU might need, that kind of stuff. And hopefully expanding that out to tribal communities in the future too, where we can start to move beyond just MSU campus and start to think about how LAMP might be useful in the broader community, and particularly in rural communities that lack basic health infrastructure, where something really rapid doesn't have to be done in a fancy dancy lab, it can be just done right in a field office, and thinking about how those types of things might be optimized for those kind of communities. But I don't want to eat up too much time before questions. I think we only have 10 or so minutes left. But
Thanks, Will, Dale, and Brooklyn. Well, let's look for questions now.
Hello? All right, so it looks like we have 2 questions so far. The first one is from Marcus Wall. He asked, what makes it so easy for the viruses in the bat populations to transmit to us?
Right, that's a great question. So it looks like bats do have a disproportionate number of viruses that can jump into humans, so we're called zoonoses. And all of these, most of these zoonoses we know of in bats, they actually use like a really conserved receptor. So if you think about what it takes for a virus to go from an animal to a human, okay, let's ignore that you have to have contact with the animal or the virus and so on, but just to cause an infection, the virus, it's got to get through a whole bunch of barriers like your skin, your mucous membranes, and so on, right? And then it's going to, so if it can actually get through all those barriers into your body, then it has to be able to get into a cell. And so that virus has to bind to the cell, usually to a receptor on the cell, and then be taken into the cell. And then it has to be able to replicate in the cell. And then it has to be able to assemble itself into its all viral packages. And then it's got to be able to get out of the cell. And when it's out of the cell, it's then got to be able to withstand your innate immune response, which is pretty good. We've got a pretty great innate, we've got lots of great mechanisms for dealing with these pathogens. So that's all very hard. And one thing that these viruses in bats have is that they use receptors. When I say they're very conserved, I mean that they're in the evolutionary tree. They're receptors that we find in almost all species. So for like Hendra, Nipah, Ebola, not so much as some of the coronaviruses, but maybe the coronavirus as well. They tend to have very wide host breadth. And why that is, maybe these viruses have very ancient origins. They've really circulated in bat populations for millennia. They've been very stable in bat populations, co-evolving over time. And we're only now just seeing them pop into other species because of these landscape changes that bring us into contact with bats or stress bats out and cause them to shed more virus. So yeah, I don't know if Will, Dale, or Brooklyn, do you have anything to add to that?
I can add just a couple things. Usually when I explain this, like when I'm giving a presentation, I do lots of what Raina just said. I also make a couple more points of like, why are bats such good disease reservoirs, right? Because I always use the contrast of like owls, because I used to study owls before I studied bats. Like, you don't hear of viruses spilling over from owls, right? Well, part of that is their lifestyle. So I always ask people to imagine, you know, owls are solitary. How hard is it for two owls to fly by each other and pass a virus? It's pretty hard, right? Well, these bats, they roost in these big 3D structures, and there's like thousands and thousands of bats just packed in there in little tight clusters. So they can transmit viruses really easy. I don't know. I mean, I always kind of think of it as like a little bit of like a cloud around a cluster of bats. So really easy for them to keep the viral transmission process going as well. And then, of course, they've got these weird immune systems that allow them to host this big breadth of viruses and possibly some really pathogenic viruses. And then I also like to say, you know, bats live everywhere. Bats live everywhere that humans live. They're so vast and so abundant. And so as we see, you know, if we put space between bats and humans, then it's harder for their viruses to spill over, right? So if we have bats living right next to humans, that makes it much easier for their viruses to transmit to us.
That's an excellent answer. Thanks, Brooklyn. You have to come along for every talk with me. They're really, really good things to bring up.
Yeah, so then our second question is from Chloe Henry. She asked, how can you determine which viruses have the ability to infect humans?
Oh, okay, that's a good one. So we're actually doing that, and we're doing it with our collaborators at Cornell and Rocky Mountain Labs. And what we're doing is we're taking sequences from the viruses we find in the field, and then we're seeing whether those viruses can enter human cells. And we do that, or when I say we, I don't do this, my collaborator Hector Aguilar at Cornell does this. What he'll do is he'll make a pseudotype virus. So he'll take that sequence from the Hendra or Nepahvirus and then insert it into another virus that you can deal with in a BSL-2 lab, just a lab like any other lab at MSU. So you don't need special containment. Then he'll see whether that virus can enter human cells and whether it can go through all of its various stages to get replicate and bud, assemble a bud and exit the cell. So that's the first stage. Can it just get into cells? Then we'll do experiments at Rocky Mountain Lab, which do require the containment, biosecurity level 4 containment, where we actually take an isolate of the virus and we'll see whether it can infect cell lines. And if it can infect the cell lines, then we will then try model species. So like, for example, a hamster is a very good model species for Nepahviruses because they get sick just like we do. We actually were going to do that. We got shut down with COVID. So we haven't done any of those BSL-4. Well, actually, we did the BSL-4 experiments in cell lines, but we didn't do them in any animals because Rocky Mountain Labs became a 100% COVID lab from January this year. They're at the front line of the NIH response to the pandemic. So we're not sure we'll actually ever do that, but that's in theory how you could do it. There has been a lot of work done in silico, meaning by computer, where people have taken, say, the sequence of the ACE2 receptor and tried to see how similar it is to, say, the human ACE2 receptor to see whether, say, SARS-CoV-2 could affect that species. So there are some things you can do in silico, but I mean, there's so much complexity in this biology that by taking a sequence and expecting that's going to tell you a virus can transmit into another species, it's way more complicated than that because there are things like immune systems. I mean, mucus. right? Mucus is really important. I was actually on a call with Cornell 2 days ago, and they study mucus in influenza. Like bird influenzas, they can't even get into our cells because their mucus, the mucus in human respiratory tract is toxic to the avian influenza. So it's got to actually have a couple of mutations to be able to survive in our mucus. So there's some really good barriers to infection. We see SARS-CoV-2. If you can get through those barriers, sometimes you can have an opportunity for those cross-species events. Like SARS-CoV-2, we understand that if you can breathe in a tiny, tiny aerosolized particle, you can then get through many of these barriers, like your innate immune barriers in your mucous membrane, so your eye, conjunctiva, and so on. You can actually get, if you can get a SARS-CoV-2 particle all the way straight down into the lungs, into the alveolar, you can then get a transmission straight into the blood system. So you actually, you bypass, the virus bypasses a few of those barriers, which is why airborne transmission of SARS-CoV-2 is such a big concern, especially to get a big dose of aerosolized particles into the lung.
Thank you. So it looks like there aren't any more questions that were in the chat, but I actually have a quick question. So like in the beginning, when COVID the outbreak happened, like I was reading up on the news and I saw that there was a lot of articles that were saying it was widely spread because of the wet markets and because of the close, frequent interactions between humans and animals. And it's also, they were talking about how wet markets have influenced past zoonotic spillovers in the past. So I was wondering if this is truly the case. What will it take to finally take into consideration proper regulation and precautions in these wet markets?
Yeah, that's a really tough one. Will, Dale, or Brooklyn, do you want to, any of you guys want to take this one?
I can start it off, and maybe Brooklyn or Dale can correct me. But it takes a lot of work, and it also, I mean, there's collaborators from Raina with the Wildlife Conservation Society that do a lot of this work about studying how these chains of transmissions actually start. So like how someone might purchase like a rat, for instance, bring that into a restaurant, cook it, and then deliver it to a customer. There's a lot of research about how that kind of stuff happens. But it's also a dangerous territory, right? Because it can lend itself to xenophobia quite quickly, because these are cultures that are very different from ours. They have very different cultural norms than ours, and it's easy to see things as, taboo, or, maybe we need to shut that down, or maybe it's something very culturally important to them. So you're right, like, maybe there is more regulation that needs to occur in wet markets, but as Raina and, Brooklyn has a preprint that's coming out now, it's more of a landscape question, I feel personally, and I think, a lot of other researchers feel too, that it's more of a landscape question about how do we manage the influx of these animals and regulate potentially like the species that are responsible, civets, bats, monkeys, things that are analogous to human immune systems and are responsible for transmission. Maybe not attacking cultural norms like a wet market, but maybe thinking more from like a landscape perspective.
Yeah, that's an excellent answer. I just add to that, in some parts of the world, bushmeat that's their only source of protein. And so there's many different contexts to wildlife markets. In all the term wet markets, like the fish markets in San Francisco, they're wet markets. So there's a lot of nuances there. It needs a lot of attention, I think, but it's certainly very complicated.
Mm-hmm. Yeah, I think that's also like a very, very good thing to think about. It's like the cultural norms and the significance of that. But yeah, thank you for sharing your perspective. I think it's just a very interesting topic because I've heard a lot of people talking about it, but it's really important to have that cultural context in it. And then Will, Dr. Lee asked, have your models predicted where we currently are at MSU and in Gallatin County with cases?
I would argue that most of the modeling we're doing in August and September was largely agnostic, more from the point of view of like some levels of like the VPR or potentially to think about how they wanted to structure things on campus this semester, how they might want to go about testing policies. They weren't necessarily from the framework of a predictive model. But interestingly, we considered a lot of parameter space where from low to high transmissibility and then across student behavior, race, if a student would actually want to come into the clinic to get tested based on the fact that, the test might hurt their nose and the fact that they might have to quarantine if they do test positive. Like there's all these sociological factors that go in. And surprisingly, like kind of our median expectations of all those things have us right around the same spot. We saw about 40 cases on campus on Friday. I think, based on most of our modeling predictions, we were sitting around that point in this time in the semester, maybe a little bit higher, maybe a little bit lower based on some of those a priori predictions. But that's definitely something we're trying to work on now with collaborators from Billings, is getting into more of a predictive model framework about how we could optimize LAMP in communities like MSU, where we know positivity is going up. We know that some behavioral restrictions are maybe not working as well as they did before, partly because of burnout, partly because, just the world itself seems to be going crazier and crazier every day. But definitely trying to step into more of a predictive framework, particularly as we try to think about how to optimize imperfect testing procedures.
That was perfect. Perfect answer. And so some of the numbers look almost like we're at the higher transmission rate, don't you think, Will?
Yeah, definitely.
Is certainly not encouraging.
All right, so it looks like, does anyone have any more questions? You're welcome to put it in the chat or unmute yourself if you'd like. Just give a moment in case there are any extra questions. All right, so thank you guys all for joining us, and thank you so much, Dr. Plowright, Will, Dale, and Brooklyn, for taking the time this evening to share your knowledge with all of us. And also thank you, Dr. Lee, for joining us again. It's awesome to see you. And so that looks like that is the end. So thank you again.
Bravo. Thank you so very much. It was phenomenal. Thank you, Dr. Plowright. Will, thank you, Dale. Thank you, Brooklyn. You make us so proud. This was phenomenal. Thank you. Bravo.
Great job. Great job.
Thank you,
Thanks so much.
everyone.
It's been really fun.
Thank you.
Have a good evening, everyone.